My name is Deandra, and I am going to be your referee for today's science battle. And so before we get started, let's introduce the participants for today. In corner one, we have Shaka, the Zookeeper X. Shaka hails from Beantown, Massachusetts, or Boston for those who don't know it as Beantown. And he went to Boston University, which is also in Boston. Uh, Growing up with seven brothers and sisters, the house wasn't crazy enough. So now he's got a snake, dog, and two cats. I guess that's why they call him the zookeeper. But they always quiet down for a home concert while he rocks the guitar. So again, let's hear it in the chat for Shaka, who will be representing vascular biology for us today. Vascular biology fans in the chat, please show some excitement. All right, I see the woos and the yays. Okay, great, everyone. And in corner two, we have LaShae, the brain surgeon Nicholson. LaShae hails from San Diego, California, and she's a beach rat at heart. After going to college in Alabama, she hopped across the pond to do her master's and PhD in Germany. While she's visited more than 20 countries, she's from the same university in Germany that originally discovered Alzheimer's disease, which is what her talk will be about today. So once again, let's hear it in the chat for LaShae, who will be representing neuroscience for us today. Okay. Now, competitors, I know you just heard the intros, and it seems like it's going to be a pretty steep competition, but I want to keep this a fair fight. So before we begin, I will let everyone take one fair jab at each other, one free taunt. We'll start with LaShae.
Hey, you're going down today. I'm going to devein your toxic masculinity.
Ooh, deveining. Oh no. Shaka, do you have anything to say to that?
LaShae, it won't take a biologist to know that your research will be a hard sell.
Oh, he brought cells into it. Uh-oh. This is going to be tough. Okay. Well, all right. Calm down, everyone. The battle hasn't begun just yet. All right. But it will be starting very soon. We are going to hear from Shaka first. Shaka, if you'd like to get ready and show us your presentation.
Yes, I'm very excited about it. Okay. All right, can you guys see my slides okay? Okay, perfect. But now I can't see you guys, so hopefully if something goes horribly wrong, someone can let me know. Okay, so my talk is about how blood flow affects blood vessels. And I hope you find it interesting. And in the background is some pictures of some cells that make up some blood vessels. So hopefully, yeah, let's get into it. So the vascular system is the system that is comprised of the heart and all the blood vessels, and it acts as like a network of tubes to bring all the important nutrients and oxygen to all the different areas of the body. So here we can see on the left a picture of a person with only the vascular system. And we can see that it's all over the body because every single part of the body needs blood vessels. You can see there's a lot of blood vessels in the brain to get you a lot of oxygen so you can think and think about blood vessels. There's a lot in the lungs to get oxygen and transport it all over your body. So it can't be understated how important it is. And the cells that line the inner walls of all the, sorry, the light's turning off. Let me just move. Okay. Sorry. It's like a sensor thing. I was gonna leave you in the dark, but and the cells that line the inner wall of all the blood vessels in the body are called endothelial cells, and they're what I study. They're very important for not just lining the inner wall of the vessel, but they're important for organizing where all the blood vessels go. So when you're growing, you need new vessels to bring oxygen to that new area. Or if you have an injury, you need to stop the bleeding, but also if something happened, you need more blood vessels to bring all the stuff that you need to repair the injury. And all of the organization that's that happens in order to generate new vessels. It's all conducted and done by these cells called endothelial cells. So that's why I think they're pretty cool. They don't need anybody else to tell them how to do it. They just know how to make a new vessel on their own. So there's two ways that the cells can kind of sense what's going on and know whether or not to make a new vessel or to stop growing or to figure out everything that it needs to do. So that way you can have a healthy vascular system. And one of those ways is that the cell can kind of like smell what's going on. So there might be a chemical or a protein called a ligand, and the cell might have a receptor and can sense that there's some signal out there, and it says, and then that receptor will bind, and then if there's that protein there, it tells the cell, okay, I'm supposed to do this thing because I just got a signal that told me I'm supposed to grow, or I'm supposed to make copies of myself or something like that. But the other way that cells can figure out what's going on around them is that they can simply just feel it. So if there's some sort of force that is on the cell, if like you put some pressure on your hand, you can feel there's a physical change there, and the cells can do the same thing. And that's also really important for their behavior. And I think understanding, for a long time, we've been looking at how the chemicals happen. So that's why we have medications that you can take. It's like some chemical that you can use to change how the cells are responding, and hopefully that can help cure the disease. But we don't have a lot of information about how the forces play a role. And I think that they play just as an important role. And I'm going to show you a particular case where the forces could be at least very important. So there's a disease called atherosclerosis. It's also known as heart disease. It's affects a lot of people. And what it is, it's a vascular disease where there's some plaque formation underneath the layer of the endothelial cells. And what's weird about this is that it's not just like any other injury that with time it will just heal. For some reason, it just, once it starts, it continues to get worse and worse, and there's no real cure for it. And we've tried, scientists for a long time have been trying to figure out what they can do to help mitigate or cure this disease. But there's something missing in our understanding of it. So why does this get worse with time? And I think I gave you guys some hints. It might be due to like the chemicals, but I'm also very interested in the forces. So maybe something about the forces going on in the blood vessel is changing, the disease and making it worse with time. So you can imagine that if without the plaques there, you have nice smooth, or what we call laminar blood flow. So you have blood going through the vessel, and there's nothing blocking it, there's nothing getting in the way. But if you have that plaque there, now all of a sudden there's a little bit of, it's harder for the blood vessels to get through the vessel smoothly, and it generates some turbulence. So the cells in the front and the back might be feeling different things, and maybe that's what's causing the disease to get worse. But in order to know, we need to test it. So that's kind of my hypothesis, is that, the forces might be contributing to the disease. So in order to test how the cells respond to different forces, we have to put them in conditions that are like blood flow. So we kind of make a little blood vessel in the lab, and we have this slide, and we put on the slide, we put some, hopefully you guys can see my mouse. I'm not sure if you can, but.
You can see the mouse.
Okay, cool. So we have this slide, and we put the cells on the slide, and we just pump, it's not actually blood, but it's just kind of like sugar water, and we can push flow over them and just see how the cells respond simply by taking pictures of them with a microscope. And what we found is that the cells align their body with the direction of the flow. So here flow is going from left to right, and we can see that the cells get longer from left to right. And here's kind of a nice video of them doing that. So we have flow going from left to right. And then over time, all the cells not by themselves, they're doing it all together, but they're all aligning. And we were wondering, why are they doing that? What does that mean for the cell, and potentially what does it mean for the disease? So before I get, so one of the things that we can do that we wanted to try is, okay, so we know that they align. But what happens if we change the flow direction? So I mentioned earlier that I was thinking, well, maybe there's some change in the flow that's happening when the plaque is there. And what does a change in flow? How does that affect the cells? And we wanted to see simply how stressed out are the cells. So a way you can tell if a cell is stressed out or unhealthy is there's this protein called P65. And you don't necessarily have to remember that, but when a cell is not stressed out, the P65 is all throughout the body of the cell. And then when the cell is stressed out, it goes to the center. And here the cells were under flow. And then we added some more flow, and the flow is in the same direction as the bodies are aligned. So here's the arrow of flow. And we can see the cells are aligning in the same direction as the arrow. But if we switch the direction of flow, so here the cells are, we flowed them this way first, and they're all aligned that way. And then you switch it to be the opposite direction that they're aligned. And we can see now P65 is in the center, meaning that the cells are stressed out. So what does that mean? So if this flow is the same direction of the cell, that's good. The cells like that. If it's in the opposite direction that its body is, then that they don't really like that and they get stressed out. So you could imagine if you kept changing the direction, or if you had turbulent flow, the cells would be always stressed out because they don't know which way it's going. So that's another hypothesis. So if this was the blood vessel, would your cells be stressed out if you have different flows because the plaque is there? I can't see the chat, but feel free to answer in the chat and I'll look later. But it does seem like these cells are stressed out. And as this gets bigger, the cells get even more stressed out. And then as it gets even bigger, because the flow is even more turbulent, the cells get even more stressed out. And then this makes the disease get worse and worse and worse. So now we think that because of the forces, we figured out, why the disease seems to get worse with time. But that leads to the next question is, but like how the endothelial cells actually sense the flow. If we figure out how they're sensing the flow, is there a way that we can make the cells stop being stressed out with changes in flow? And then maybe it gives the body time to heal. So we know that there's some proteins in the cell that are important for the cells to align. So there's this protein called V-cadherin, and it's along the edge of the cells, and the cells use it to kind of pull on each other and talk to each other. And here's an experiment where, this little mouse can't move, but an experiment where the cells are subjected to flow. This is no flow, and then this is after about overnight with some flow, and we can see that they're aligning in this direction. But if we don't have this protein, if we remove it from the cell, we can see that the cells, even under flow, they can't align anymore. So, and like I mentioned, this protein is along the border. So in green is actually an image of where this protein V-cadherin is in the cell, and we can see in static and in flow conditions that it's all along the edge of the cell. So it means that the cells are probably communicating with each other saying, hey, we should all go this direction, or we could all, we should stay this way. So if we can figure out maybe how this protein is working, then maybe we can figure out a way to make the cells feel better even when there's turbulent flow. So another interesting thing is that we found that these proteins are actually activated where there's less flow. So here's a big artery that's connected to the heart, and it's called the aorta, and it's kind of curved. And you could imagine, so this is kind of like looking at it sideways, and the flow would go from here and then down. And as the flow goes up and around, it's pretty smooth and constant. But around this bend, it kind of gets like the blood flow kind of gets shot off to the side. And there's a little bit of turbulent flow here just because the vessel is curved. And we can see in this area of turbulent flow that there's this activation of V-cadherin. And what's really interesting is that this is the spot where we also see atherosclerosis in these plaques. So this is another picture of the same thing. It's a side of the aorta, and the blood flow would go like this. And on the top where there's nice and smooth flow, it's nice and thin, and it looks like there's no plaques here. But all along this side, we can see all these plaques. So there's definitely something interesting going on with V-cadherin and turbulent flow. And in this case, it led to atherosclerosis. So if we can change how V cadherin acts in response to flow, can we help prevent the disease? So I'm thinking to myself, what are the things that regulate how V cadherin is responding? And doing some research from some other scientists, we found that there's this protein called LGN, and it act and change how V cadherin acts. If I could change something that affects it, I can hopefully find some more useful information. So I decided to see how LGN affects how the cells sense the flow. And I did that in the same way, just by removing LGN from the cells, like I showed you with V cadherin earlier. And when I remove LGN from the cells, we can see our cells that have LGN, they're on the top, and the cells without LGN are on the bottom. And these kind of colors show you how well aligned the inside parts of the cell are. And we can see with the direction of flow. So the flow is going from left to right. And if it's more blue and green, that means the cell is more aligned. And if it's more pink and purple, that means the cell is less aligned. And here we can see that without LGN, the cells don't align that well. And this is what V cadherin looks like. So we can still see it's at the border, and you know, it's there as it should be, but it's just behaving a little bit differently. And then the last picture is just all these pictures kind of put together so you can see the full body of the cell. And what's really interesting is you can notice that all these cells are still long. Like, they don't look like the static where they're all kind of not really aligning. The cells in both conditions are long, but these guys are going in the wrong direction. So this graph on the right is just showing that if you quantify how long the cell is, they're the same length, but for some reason, these guys are confused and they don't know which way the flow is coming, indicating that, you know, maybe this protein is important for how the cells can sense the direction of flow. So the next thing we wanted to do is simply see where this protein LGN is located. So this full body is a cell, and the white and gray is where LGN is. So we can see there's a lot in the center here, and there's a little bit all along the body. And then when we put 5 minutes of flow over the cells, we can see kind of the LGN, it's a little bit less throughout the rest of the body, but it goes to the edge where we know that V cadherin to V cadherin to tell it to do something. And then after the flow is stopped, it goes back. So maybe this protein is going to where it needs to go to tell the cell to go in the right direction. And if we can maybe find a way to manipulate it and kind of get it to go where we want, then maybe we can make the cells feel happier in a situation that would stress them out. But we still don't know how the whole thing works. There's a lot of work to still be done. So that's kind of what my job is now. But maybe if we can figure it out, we can find a way to prevent the disease from getting worse. So thank you. This is everybody in my lab. And yeah, let me know if you have any questions or ideas, because this is a work in progress. So if someone in the audience noticed something, I'd be happy to look into it. Thank you.
Thank you so much, Shaka, for an incredible talk. And now we'll hear from Heather to explain how the voting is going to work for this event.
Awesome. Thanks, Deandra. So for the voting, you should get a little voting poll launching up on your screen right now. And we're going to vote on 3 different things on Shaka's presentation. So how understandable was the presentation? Were you able to follow most of what Shaka said? If he used any big or new words, did he explain it well? So if you were completely lost, vote 1. You understood everything, vote 5, or you can vote anywhere in between. We also want to know how much did this presentation spark your curiosity and imagination? Even if you don't want to do this research yourself, how much did it make you think, wow, that's cool? And lastly, how much more do you feel you know about this topic compared to when you joined the Zoom meeting? We want to get an understanding of how much you've learned. If you've learned a lot, a little, or nothing at all, just let us know.
All right, and while everyone's voting, we are going to ask Shaka some of the questions that you've asked in the chat. And so, feel free to also ask any more questions if you have any. But Shaka, once again, great presentation, great presentation. I'm sure everyone else in the chat agrees, and we got a lot of questions. One question that we got, you were talking a lot about stress. And you're talking about the stress of these cells.
Mhm.
One student was wondering, is this somehow related to like excessive stress induced heart failure? So is the stress on the outside, like, when you're like stressed about an assignment, is that also related to the stress in the cells?
Um, Sorry, could you repeat the second part?
Absolutely. So is the stress that you're feeling from like, I've got to do this homework, I've got so many plans, is that kind
Okay.
of the same sort of stress that we're talking about with these cells?
So yes and no. So yes, so we're looking at a type of stress called inflammation. And inflammation is usually something that happens when like you're sick, or in a case like you're stressed out about homework or something like that. And your body releases all these chemicals to say, we should be on alert, or we should respond to danger, whatever that danger may be. So they are definitely linked together, but it's not like being stressed about your homework will cause your endothelial cells to be stressed out in this type of way. So if you're stressed out, it can lead to more inflammation in your body, which isn't ideal, but it's not the exact same thing. It's not that, yeah, it's a specific type of stress, not all stress.
Okay, I think that answers it for me. Thank you, thank you. We also got a lot of questions in the chat about the disease itself. Specifically, a lot of questions about survivability of this disease. Is this a degenerative disease? Have people lived from this disease? What's known about that?
So unfortunately, no. This is one of the leading causes of death in the United States, especially. It usually happens over a very long time, and there's a lot of things that you can do to prevent it. So the first plaque might form 40 years before you actually end up needing to go to the hospital with something. So that's why doctors are often very always telling people to exercise and eat a healthy diet to prevent the plaques from ever getting there in the first place. But yeah, no, oftentimes it's unfortunately very sad. There's not much you can do. It can lead to various different things like the heart disease, heart failure, as well as stroke. So these are things that unfortunately we hear about too often. And right now, there's not really too many cures.
Oh, it's a good thing that you're working on it then. um, Another question that we had to do with the cells themselves. So how do the cells really have this sense of touch that you were talking about?
Yeah, so it's actually really interesting. I mentioned that there's chemical and physical forces, but actually what the cells do is they have these proteins called mechanosensors, and it's just what it sounds like. They're mechanical sensors. And what happens is if there's a force on the cell, then these proteins release chemicals into the cell that they can respond to. So one of these things might be calcium. If you push on the cell, it releases calcium, because there's these little sensors that feel that they were pulled, and that tells them for calcium to go into the cell, and then that tells the cell what to do. So really what the cell is doing is converting the physical input into some sort of chemical signal that it can understand. Hopefully that makes sense. But that's actually what happens when you feel anything. Your body's converting that into chemical signals that your brain can understand, and the cells do that their own way.
Okay, I think I'm getting it now. And so I think we're going to have time for one more question. If you haven't voted yet and you'd like to vote, this is your last chance to vote for Shaka. While you're doing that, I will ask the last question. one of the students in the chat was really interested by the experiment that you were doing that you showed with the sugar water that was kind of colored red like blood. And they wondered if it was possible to try the experiment at home. And if not, can you describe like how difficult it is to set up something like that?
It's actually, I don't think you could do it from home just because I don't know where you would get the cells from. We get them actually from people that donate them, and they have to undergo a procedure in order to get them. So we can only get them from a licensed doctor. And so it's not something you can just get by yourself. It's something that we're lucky enough to have doctors that are able to do that and then give them to us to do the experiment. But it's just something that you can kind of keep in mind, that every time you feel your heart beating, it's pushing blood over these cells and it's letting the cells know what to do. It's more or less, you're always doing the experiment in your own body.
Okay, so you hear that everyone's already doing the experiment at home, so
Yeah.
no need to set anything fancy up. Okay, thank you again so much, Shaka. We are now going to give the floor to LaShae, who will talk about neuroscience for us today. So let's hear it in the chat for LaShae.
Thank you. Can you hear me?
Yes, we can hear you just fine.
Okay. So I will share, and then I will make this. All right, great. All right, so it's nice to be able to give a talk to you guys about why vessels matter in neuronal diseases. However, before I go into this, I just want to share two important key points. So the first is that vessels are a lot like neurons. So as Shaka said before, that blood vessels and neurons occupy every portion of the body and every organ. And then at the cellular level, both neurons and vessels actually have the same cellular features. So for example, here you have what's called the neuronal axon, and you have blood vessels. And at the tips of these cells, you have what's called these filopodia-like structures, which are actually finger-like structures that allows you blood vessels and neurons to sense their extracellular environment in response to attractive cues and also repulsive cues. All right. Thing I want to introduce is Alzheimer's disease. So Alzheimer's disease is something that I'm working on for my research. And Alzheimer's disease is a neurodegenerative disease in which you have a severe loss of nervous tissue within the brain, as you can see here in his Alzheimer's disease brain, in comparison to a wild type normal healthy brain. And just to give you some quick facts about Alzheimer's disease in the United States. So it is also something that affects nearly 6 million people within the US. And this will actually become 13 million people within the next 20 to 30 years. And unfortunately, over the past 2 years during the COVID-19 pandemic, there's been an increase in AD-related death. And also, 1 in 3 seniors also die from Alzheimer's disease or some other form of neurological disorder. And Alzheimer's disease is also a really expensive disease in which $355 billion are used on patient care, in which is provided by 11 million people over the course of 15.3 billion hours with respect to people taking care of their loved ones who are suffering from Alzheimer's disease. So because of Alzheimer's disease being so prevalent within the United States, it's very likely that you, as you get older, will either succumb to Alzheimer's disease or might have a loved one who's also suffering from the disease. So the way that Alzheimer's disease works is that there's a protein called amyloid beta, which under normal circumstances forms these single monomers, which are actually healthy for neurons, which promote neuronal growth and neuronal function. However, these monomers can aggregate over time and form these protein clumps called, which are toxic amyloid beta oligomers. And these oligomers are responsible for causing neuronal or abnormal neuronal signaling, leading to neuronal death. And the way that this works is that under normal healthy circumstances, you have synapses, which are the communication points between two neurons, which allows for signals to pass one neuron to another via receptors, which allow for normal neuronal signaling and function. However, in the case of Alzheimer's disease, you have the formation of these amyloid beta oligomers, which become very toxic and interact with these same receptors, which allow for abnormal or toxic signaling within a neuronal cell. And in response to this abnormal signaling, you have what I call I'm sick signals, which are signals that tell cells, neurons that they are not healthy, which are actually tagged at the synapses. And then what you have are immunal cells. So these are cells that police the brain. They are responsible for removing unhealthy cells and toxic waste. And in response to these I'm sick signals, they begin to attack these unhealthy neuronal cells, leading to synapse loss and eventually neuronal loss. However, there is a way that neurons can survive in response to these toxic amyloid beta signaling. So recently, or actually before I came to the lab, my colleagues discovered a drug which they found that interacts with this, these receptors that block this toxic amyloid beta signaling, preventing this abnormal signaling. And via this drug, therefore, you can protect the neurons and prevent this immunocell attack. So even though you still have the presence of these toxic amyloid beta oligomers, you still allow for normal neuronal function and signaling. And this you can also see in tissue. So what I have here is an example of an image from an Alzheimer's disease brain, in which you can see the glial cell marker or immune cell marker GFAP, which is in green, and an SV2A marker, which is a synapse marker, which is in pink. And in the Alzheimer's disease brain that doesn't receive this drug, you have the immune cells in green attacking these synapses. So you can see these purple dots within the green cell. However, with the drug treatment, these cells are no longer attacked by these immune cells. So you see no pink dots within the green cells, meaning that these cells are healthy. And although you have amyloid beta, they're able to function normally. All right. So I spent a lot of time talking about neurons and Alzheimer's disease. However, I want to remind you that blood vessels and neurons are alike. So what about blood vessels? So blood vessels, they supply oxygen and nutrients to the brain. And because blood vessels and neurons are very much alike and they respond to similar signals, they are very close to each other. So no blood vessel is more than 10-20 microns from the nearest neuron. And like neurons, vessels have these filopodia, so these finger-like sensors, which again sense the extracellular space, and they can respond or grow towards or move away from these extracellular signaling molecules. So for example, blood vessels can grow towards regions where they can sense low oxygen. And the process of blood vessels growing towards these signaling molecules, this is a process called angiogenesis, in which you have the formation of new blood vessels from existing blood vessels. All right. So how do we study vessels in Alzheimer's disease brain? So in the lab, what I do is I work with a mouse model in which I have a normal, healthy, wild-type mouse. And then I also have an Alzheimer's disease mouse. And I let these mice age over the course of 12 months. And then at the 12-month time point, I collect the brain tissue, I make brain slices from the tissue, in which I then label these tissues with different antibodies, which mark immune cells, blood vessels, and amyloid beta cells, or amyloid beta oligomers, which I then image. And as you can see here, this is one image from a healthy brain. And in this healthy brain, you can see the vessels, which are marked by this vascular marker called lectin. And these vessels are healthy and normal, and there's no presence of amyloid beta. However, in the Alzheimer's disease brain, you have these vessels which look smaller, less vessels, and they look somewhat unhealthy, as you can see here. And that these vessels are abnormal in regions where you have these amyloid beta plaques. All right, so what's going on, or what's happening to these blood vessels in the Alzheimer's disease brain? So again, under normal conditions, you have very healthy blood vessels, which function normally. They supply oxygen and nutrients to the brain. However, in the Alzheimer's disease brain, you have the formation of these amyloid beta oligomers, or these dense plaques. And these plaques, they actually take up space and they move vessels away from each other. And then what happens is that you have areas where there's very low oxygen. And in the presence of very low oxygen, the surrounding tissues secrete these I'm sick signals, or there's low oxygen signals, in which you produce or you promote angiogenesis, so the formation of new blood vessels. However, because you have a lot of these I'm sick signals being produced, you have what's called abnormal angiogenesis. So hyperangiogenesis, where these blood vessels are forming so quickly, but they actually don't form proper blood vessels. And because of this, over time, these blood vessels become sick, and which again attracts these immune cells, which actually attacks and kills these blood vessels. All right. So with respect to the title of my talk, why are blood vessels important in neuronal brain diseases? It's because neuronal diseases are actually really complicated. So although this is a neurological disorder in which neurons are actually being impacted and there's a lot of neuronal death, there's actually a lot of other cells involved, namely blood vessels, immune cells, and a lot of other cells that I haven't even mentioned today. However, there are drug treatments that are available for neurons that are neuroprotective, that prevent the neuronal loss. However, because neurological diseases are complicated, you need drug treatments that also protect vessels as well. So even though you have the same neuronal disease, you have multiple cells involved, which needs multiple strategies to find a cure. So in this case, what I'm using in a lab, I'm using mouse and human and cell culture models. I'm using imaging. We do a lot of behavioral testing in animals. We also do gene expression profiling in the hopes of, again, finding another drug that can not only be protective of vessels, but also protective of Alzheimer's disease, which we can hopefully find a cure. And having said all of that, I want to leave you with my favorite quote from my favorite scientist, which is, everyone is a neuroscientist because everyone has a brain. And I love saying this, and I love telling this to students, because no matter what your background, no matter what your interest in, for example, I'm a neuroscientist who's also interested in blood vessels, but I also am interested in computer science, which is also something I use to study the brain. So there's something that everyone can do that can contribute to the field of neuroscience based off your own interest. So thank you for listening. I hope you understand everything in my talk, and I will be happy to answer any questions. Okay.
Thank you, LaShae, for an incredible talk, and also thank you for that quote. I've had a brain, so I really didn't know I was a neuroscientist. So thank you so much for that. Just like with Shaka, Heather is going to put up a poll, and this time you're going to answer those same questions for LaShae. So while I am asking questions, feel free to use your nice healthy brains to vote for LaShae's presentation. Okay, so we also got a lot of questions during your presentation, and I will ask a couple of them here now. One question that we just got in the talk was actually about you mentioned that you use a lot of different strategies to study this disease. One being like looking at human brain and one looking at mice brain, the brain in mice. Are there any differences between those two different types of brains that could affect your research that you're doing?
Yes. So mouse brains and human brains are actually similar in a lot of aspects, in a sense that they both have blood vessels and neurons, and they have the same signaling molecules. So this allows us to use animal models in order to study human conditions. However, there are definitely differences between humans and mice in terms of where certain genes or certain things are expressed within the cells. And then also Alzheimer's disease itself is also very complex, in a sense that you have the disease that's related to genetic disorders, some mutations, there's some other different signaling molecules that contribute to Alzheimer's disease, which can't be reflective in the same mouse model. So although you can use an animal model or a cell culture dish in order to study the human condition, you always have to go into a human model or work with human patients in order to find an eventual cure for the disease.
Okay, so it is a model. It gives you a lot of insight, but you do have to look to humans for the final thing.
Yes, definitely.
Okay, thank you for that answer. The next question we have is somewhat related. So we had a question about I guess how you actually got the pieces of the mice brain to do the experiments. So is this done by X-ray? Is it a surgery? How does that work?
All right. So actually, there's different ways you can do this. So in our lab, we have methods in which we age the mice, and unfortunately, we have to sacrifice because we remove the brain from the mouse. However, there's also other techniques as well. So we have what's called PET imaging. So this is in vivo imaging. So you can monitor the brain in a live wake animal, again, under normal and wild type conditions as well. And then there's also things where you can do in vivo imaging, where you can use a microscope and you can actually look at very precise, very precisely at the movement or functions of cells, again, in an alive wake animal too. So there's, again, there's different methods and there's different strategies. What's nice, or the good thing about harvesting the brain tissue, is that you can slice the brain in several different sections. So that means you can use the same brain to study different proteins, different cell types. Yeah, that you're interested in as well.
Okay, and I think we have time for one more quick question. So we know Alzheimer's is a disease of the brain. And does this disease, is there a way for this disease to only affect the brain and not the rest of the body? Because I know you mentioned a lot about like the vessels and everything like that. Is this also affecting like other vessels in the rest of the body, or is this kind of really localized? And if so, is that a way to kind of make the disease not be as fatal, I guess, if it's only affecting one part of the body is, I think, what the student was getting at.
Yeah. So Alzheimer's disease is a terminal disease because, again, we as humans, we need our brain to function. So if we have a lot of neuronal loss or a lot of brain loss, this can impact the quality of life and also lead to death. Within the brain, we have something called the blood-brain barrier. So the brain has its own protective barrier around it that prevents things within the peripheral nervous system impacting the central nervous system. And this blood-brain barrier is also established by blood vessels as well. And actually, one reason why you have the accumulation of these amyloid beta plaques is that these toxic molecules are not able to pass out of the blood-brain barrier and to be safely removed from the body. And so Alzheimer's disease is something that is specifically something that happens within the brain. But it's also because there's an improper function of the vessels as well that's preventing this removal of these toxic molecules from the brain.
Okay, great explanation. It seems that we are closing down the voting now. Thank you again so much, LaShae. That was incredible and very intelligible responses to the questions as well. And I think I am now going to pass it on to Heather, who has collected all of your votes and is going to crown the winner for us tonight.
Hey, everybody. Let's see.
I can stop. Okay, great.
All right. So thanks, everybody, for coming to this super science showdown. And congrats to Shaka and LaShae today for their amazing presentations. But you guys are probably really interested in who actually won the battle. How do your votes stack up? Can I get a drum roll, please? All right. So for the first question, we asked how understandable the presentation was. And here we have LaShae in the lead by just a few fractions of a point. I have a feeling this is going to be a close one. And for the next question, we asked, how much did this presentation spark your curiosity and imagination? And again, LaShae is leading by just a little bit. These are both really great presentations, and you guys rated them both very highly. And for our last question, how much did you learn? How much did you feel you know about this topic compared to before? And these scores are dead even down to like 4 decimal places. They're both like 4.3642 or something crazy. They're dead even, exactly the same. I've never seen anything like it. So we averaged all the scores together, and you guys can probably guess who won, but LaShae won by just a few fractions of a point. Her presentation was just a little bit more understandable, a little bit more interesting to you guys tonight. So I'd like to crown her the winner. Congrats, LaShae.
Yeah, thank you. But Shaka also gave a good talk as well. It was really interesting.
For sure. It was a close one. And just so you guys know what's coming up in the next few weeks, Maya said this earlier, but today we did a round of Flip Science Fair Versus. Next week, we have a demo, hands-on demo day. The registration for this is already closed, but if you guys signed up for it, you and your parents would have already gotten emails about it. So we hope to see you next week if you already signed up. If you're not signed up for that, in 2 weeks, we're gonna have another battle. We have 2 great scientists coming your way. And the week after that, we're having a discussion about what does science do in policy and how does that influence our lives day to day with our mayor, Justin Elliker. So we hope to see you guys soon, and thanks for coming to this battle.